Hello, everyone. Welcome to our Precision Medicine Startup Showcase. My name is Stephen Collins. I'm the CEO of Matter. Our last Startup Showcase in September was focused on solutions for life sciences companies. This one is focused on precision medicine. The evolution from basing diagnostics and treatments on population norms to basing them on an individual's characteristics is one of the most important trends in medicine. And in the future, doctors will routinely have at their disposal sophisticated analyses of large amounts of data from numerous sources to assess what treatments to give or recommend to a particular patient, enabling physicians to select targeted treatments with higher rates of success and lower likelihoods of adverse effects. For today's showcase, we've invited 3 Matter member companies whose solutions harness genetic information and other data sets to improve clinical decision-making and mitigate complex disease progression in oncology, in hypertension, and in pancreatitis. We'll hear from AIQ Solutions, Ariel Precision Medicine, and Geneticure. Each company will have 7 minutes to present their solution, and then we'll take 7 minutes of questions from our panel of experts who come from 3 of our partner organizations. First, we have Eric Messner, who's the Senior Vice President of Business Development at Paragon Biosciences. I very much appreciate Eric's resume because, like me, he has an MBA from Kellogg and he spent time at Abbott Laboratories. He's been with Paragon and their portfolio companies for the last 4 years. Paragon is a Chicago-based firm that builds and invests in life sciences companies with focuses in AI, gene therapy, synthetic biology, and biopharma. Thank you, Eric, very much for joining us today.
Thank you for having me.
Second, we have, we have Mayank Taneja, MD, who is Director of Venture Investments at OSF Healthcare. OSF is a highly innovative health system based in Peoria that we've partnered with since we first opened. Mayank joined them about 6 years ago and is in their venture group. Thank you, Mayank, for joining us. And third, we have Ethan Wergelis-Isaacson, who is Associate Director of Digital Health Strategy and Business Development at Utsuka. Utsuka is a Japanese pharma company that has 3 kind of identified focus areas, which I find particularly interesting. They identify neuroscience, nephrology, and digital innovation as their 3 areas of focus going forward. Ethan just joined Utsuka last month. Before that, he was running the health tech vertical of Dreamit Ventures. So thanks very much, Ethan, for joining us. Our first company is AIQ Solutions, and to present will be Eric Horler, who is the president and CEO.
Thanks, Stephen. Let me get my slides going. Great. Well, thanks very much for having me. As Stephen mentioned, I am the CEO of AIQ Solutions, where we're developing a software platform to help improve outcomes for patients with complex diseases like metastatic cancer through therapy optimization. So you're going to hear about different approaches to this general concept of how you tailor therapy to the specifics of an individual patient. AIQ is a little different in that we come in the point in care after the patient has already been diagnosed with the disease and after initial therapy has already been selected. And we look at situations where the outcomes that you get from that initial therapy selection don't always meet the expectations. One of the reasons for this in a complex disease is that not all sites within an individual patient respond the same to treatment. This is known as intra-patient treatment response heterogeneity, which is a lot of big words. But fundamentally, it's very difficult to understand with these existing technological solutions. And more importantly, it can really limit the effectiveness of therapy. We've been able to show and publish with our technology that having less than 10% of individual lesions resistant to a therapy drive overall poor outcomes for the patient. So it's a little bit of a change in thought process from trying to maximize an overall response for a patient to instead trying to identify and treat resistance and minimize the level of resistance. And this is where AIQ's technology comes in. We have developed a software platform that automatically identifies all of the sites of disease within the patient, quantifies the disease for each individual disease site or lesion, and then classifies them based upon whether they are responding to treatment or resistant to treatment. We then use this information to make predictions as to how this patient is ultimately going to do on the chosen treatment. At the same time, we identify healthy organ systems that are at risk of toxicity, because especially in the case of oncology, every treatment decision is a balance between expected effectiveness and expected or risk of toxicity, and provide these predictions to the clinical team as well. So how does this work? So you start by taking imaging data from multiple points in time and upload these to our cloud-based system. It's an entirely cloud-based software as a service business model. We use our proprietary technology to first identify the regions of interest within the patient, and then we use artificial intelligence to determine which of those regions of interest are malignant disease. Once we've done that, we use our patented technology to match images from multiple points in time with enough precision that we can then quantify how each individual lesion is responding to treatment. There's not any other technology that is able to do this at the individual lesion level. We take this very rich data from all of these individual lesions, feed it into a deep learning model, and use that then to predict how the patient is going to do. And a very similar process is done for the toxicity assessment as well. So let's think about what this looks like in clinical practice. Let's imagine a patient that has cancer that is metastasized to her liver. She's going to get a baseline scan right when she starts therapy. And then shortly after therapy, typically just a few weeks, you get a second scan. This information is sent to AIQ. We perform our analysis, and one of the things we return is what we call the AIQ score. This is a biomarker. It's a number between 0 and 1 that indicates the likelihood that this therapy is going to have the optimal effect for the patient. So let's imagine the scenario where that number comes back predicting a suboptimal response for this patient to therapy. The next thing that the clinical team is going to want to know is that suboptimal response being driven by widespread resistance? So if you look at this liver at the top of the slide, you'll see that almost all these lesions are red, indicating widespread resistance for this patient. This is a patient who's really a candidate for a change in approach, either a different drug or, depending upon the situation, perhaps palliative care, and avoid the time and toxicity of an ineffective treatment for many months. On the other hand, if this poor prognosis is driven by a limited amount of resistance, in the case of this liver, you'll notice there's only 2 lesions that are red. All the others are either green or gray. This patient is a candidate for some sort of localized ablation procedure, could be radiation therapy, could be a surgical procedure, to remove the resistant disease before it can clone and spread, and then keep the patient on a therapy that is by and large still working for that patient. Now, if the prognosis comes back positive, in many cases, you're just going to continue therapy. But in some cases, you can actually use our technology to de-escalate therapy and avoid the unnecessary cost and toxicity associated with potential overtreatment. AIQ initially started with a technology that focused on just quantifying whether or not a therapy was effective. We've moved beyond that, and today we're doing quantification and prediction. We're looking at both effectiveness and toxicity, and we're applying this to a broad range of cancers using multiple different imaging modalities as our input. But more importantly, where are we going? So we're now in our early research moving beyond oncology. In particular, we have some encouraging research into neurological disorders, and we are now starting to look at how we bring non-imaging data into our algorithm. So this could be things like blood-based biomarkers or pathology data that can help us make a more robust assessment and prediction of this patient's treatment effectiveness. Globally, we see a total addressable market of about $7 billion for technologies like ours that are focused on treatment response for complex diseases. In the US, the number is a little over $1 billion. Our primary competition is what's in the lower left-hand corner or lower left-hand quadrant of this image, which is standard of care. This is basically manual analysis and assessment of lesions by a radiologist by standard. That's going to be 3 to 5 lesions only. If you move up into the upper left-hand quadrant, looking at companies that are applying artificial intelligence to imaging data, there are a large number that are doing so for the purpose of diagnosis, but this is very different from what we do. There are some companies that have automated portions of the radiology workflow. None of the companies in the upper left-hand quadrant, however, can assess treatment response for each individual lesion, nor make the predictions around effectiveness and toxicity that we can. On the other hand, in the lower right-hand corner, you have the companies that are looking at biomarker data to make predictions of treatment response. They lack the spatial information that is really critical to identifying and then attacking resistance. This is what makes AIQ unique. We are right now in what I would call the build phase, focused on generating evidence, securing reimbursement, and establishing the IT infrastructure that we will need to interface with hospital systems. By early 2023, we expect to be ready to move into the growth phase and really invest heavily into marketing to provider systems. So thank you very much for your time, and I'm happy to take questions.
Thank you so much, Eric, for your presentation. We'll turn it over now to our panelists to ask some questions.
Eric, it's Eric Messner. Thank you for your excellent presentation. I just want to make sure I understand the technology before I start asking questions. You have to have at least 2 images, right? And you compare the lesions from time 0 to time 1. Either they stayed the same, they've grown, or they've shrunk. And then you assess the effectiveness based on that change over time, correct?
That is correct. Yeah, so we're focused on longitudinal data. So you do need to have at least 2 time points in order to figure out the change.
Okay, and does your software handle, or will it handle multiple images over time, like 3rd, 4th, 5th, 6th, 10 images over time?
Absolutely. And so what you can do is you can just do every single permutation of comparison there. And just as a random example, we did one study with NIH where they sent us patients that had 20 scans over the course of treatment. And we looked at all those different permutations to try to see what's going on over the course of that treatment. But yes, as many scans as you have, you can learn something.
Okay. I guess my first question is just a basic question, and that is, I'm assuming that you'll have to either get a de novo clearance or a 510K clearance from FDA. Is that part of the business plan?
So we already have a 510K clearance for the technology. So it can already be used for patient management today.
Okay. And I guess the follow-on question is, who is the customer? Is it the radiology department? Is it a cancer treatment arm of a hospital? Or who is the customer? So
we see the clinical champion. I'm going to answer your question slightly differently, because as we all know, when you sell to hospital systems, there's a lot of stakeholders, and you really have to be able to appeal to all the different stakeholders. We see our clinical champion as being the medical oncologist. It's really a medical oncology planning system. That said, we are working with radiologists. There's no question they're important stakeholders, but we don't see them as the champion or the decision maker.
Eric, this is Mayank. Thanks again for the presentation. Can you just talk a bit about the validation that has been done around this software, and then how did you get to these color codings that you have, and what supports that work?
Sure. So in terms of the validation of our ability to identify the malignant disease and then to classify whether or not it's resistant or responding, we did this with a panel of radiologists annotating scans. We looked at somewhere between 1,000 and 2,000 different lesions in that data set and used that to train the artificial intelligence engine to be able to determine whether or not that cancer is malignant, or whether those regions of interest are malignant cancer, and then also to look at the classification.
Got it. And just to follow up, what was the result of that? What did you find out? What were the findings?
So, not unusually when you're looking at artificial intelligence versus radiologists, our predictions were more consistent than the variability among the different radiologists. If I recall correctly, we had a panel of 3 radiologists that were all looking at the same data. In terms of specificity and sensitivity of the tool, both are over 90%.
So what claims specifically did you get from the FDA?
So that is a very poignant or a question. Right now, the claims we have in our 510K are around the identification of regions of interest and the quantification of different data about those regions of interest. The predictive portion is something that we probably will have to go back to the FDA to make as strong of claims as we would like. That also is consistent with, I mentioned in my one slide that we're in the build phase and we're working through the reimbursement process. Part of that does involve generation of outcome data. Our strategy is to go back to the FDA, talk to them about the outcome data that we're generating and see whether we can get a more aggressive claim around the prediction piece of our technology using that data.
Mm-hmm. Is there like an 80-20 rule as to kind of the outcomes after the initial treatment end up being optimal, suboptimal, limited resistance or not? Is there like a majority versus not?
Boy, that's based upon the clinical applications that we've seen. It's not 80-20. There's about 60% where there's an opportunity for optimization, about 40%. And I'd have to go back to the data to say 100% for sure. These are the numbers that I'm remembering off the top of my head, where the answer is just, you know what, this therapy is good, stay the course. But even within that 40%, if you take prostate cancer as an example, if you can determine a patient that can stay on first-line therapy, say a singlet drug versus moving on to a triplet therapy, which is becoming more and more standard of care, the cost savings over a 6-month period of time are $30,000 to $40,000. So even in those patients that would do well on the chosen therapy, this opportunity to potentially go with something less aggressive has real benefits for both the patient and for the system.
Eric, you mentioned before that you can use the software to predict outcomes with treatment. How do you handle the situation where if a lesion is not responding and a recommendation is made to switch, how does the software handle potential progression of the disease once the chemo agent is withdrawn? Is it learned enough to know that a cancer may progress more quickly where it's only partially regressed and now it would progress? Like how do you handle removing a drug, or how does the AI handle that part of it?
So right now, we don't have the analytics. If I can understand your question correctly, you determine that a patient has got resistance to therapy A.
Yes.
You're considering switching to therapy B. And the concern is, might therapy B actually have resistance in some of the disease that was responding to therapy A? So could you potentially be trading off something that is worse than what you had before? Right now, the technology doesn't have the ability to look at these specific therapies and predict that. What we would do is as soon as you switch the patient to that new therapy, shortly into that therapy, do another treatment response assessment and see how that second therapy is working.
How do you price? Oh, sorry, Stephen.
Well, go ahead, Ethan. One last question.
I'm interested in your business model. How do you price?
So the business model is software as a service. The revenue we're generating today is with pharmaceutical companies who are using the product in clinical trials. We're doing that because we don't have the reimbursement in place. A lot of around what we're going to be able to get for pricing is going to depend upon what we're able to get a payment rate when we do get reimbursement. Looking at some analogies in the marketplace and also looking at our health economic model, we believe that we can justify a price per analysis that's somewhere in the $500 to $1,000 range, but we're going to have to go through the process and see whether or not we can get those kinds of payment rates from the payers.
So it's
Eric, thank you so much for your presentation. Sorry. Thank you so much, Eric, for your presentation. Thanks to panelists for their questions. We will move on to our next company, Ariel Precision Medicine. Co-founder and CEO Jessica Gibson will be presenting.
Thanks so much, Stephen, and I appreciate everybody having me here today. As Stephen mentioned, I'm Jessica Gibson. I'm CEO and founder of Ariel Precision Medicine. And our vision is to really be a global leader in facilitating an earlier diagnosis, targeting treatments, and long-term management for digestive and metabolic diseases. We've developed a proprietary platform that is enabling us to do this. We generated $1 million in revenue last year for our first solutions applied to the complex disease of pancreatitis. We're looking at over 30% growth this year, and just appreciate the opportunity to share with you some of the problems that we're addressing right now and where we're heading in the future. So I'm sure, as many of you are aware, that complex diseases are really bankrupting our US healthcare system. Almost 90% of healthcare spending goes towards managing end-stage chronic disease. And with the opportunity of precision medicine, we can use early signs and symptoms, genetics, environment, lifestyle, and events to predictively model how a patient is going to progress, to mitigate a lot of these challenges that we face in a diagnostic journey when we're looking at symptoms or features of a disease to diagnosis. And this is really where when advanced cancer presents, when you've got a chronic irreversible condition, we've missed the opportunity to prevent that progression, and also an incredible expense to the healthcare system. So I co-founded Ariel with Dr. David Whitcomb, who is a global expert in digestive and metabolic diseases. He was director of the GI Hepatology Nutrition program at UPMC for almost 18 years and brings a medical advisory board of global experts. And internally, we are leveraging expertise from biomedical engineers, computational biologists, AI specialists, and geneticists as well. So this is really the problem that I want to be able to provide some more insights into today. The way that we define a disease is feature-based. We have a spectrum of symptoms, and they're kind of pulled to the average, where the symptoms and phenotype is used for a diagnosis. And this is failing complex diseases, and I'll use diabetes as an example. Diabetes is usually identified with elevated blood sugar. But the reason that this is insufficient is that there are actually a variety of different mechanisms that in isolation or combination can drive different patients' disease. So when we use pancreatitis as an example, it is one primary presentation, but a variety of different causes, and the treatment has to be able to be aligned to the mechanism. We need to be able to move from treating symptoms to treating the underlying biologic etiology of disease. Now, when you think about genetics, I would say one of the approaches that we're taking that's really different and kind of countercultural to the big data approach is that genetics are complicated. Patients can have a variety of genetic variants that impact the way that their body functions or doesn't function. And it's not agnostic to the patient condition. So the genetic information doesn't change over a patient's life, but everything else in their environment does. Their age, lifestyle, events, and medications that can be taking can provide variable outcomes into a way a patient's disease starts and progresses. And there aren't really enough people on the planet to power a big data approach alone. So the approach that we're leveraging in our advanced platform is essentially virtual biologic models of an organ. When we look at the cell types, so we can take multiple variables of varying effect and model it under a variety of conditions in a single patient. And this is, as Stephen was saying, moving from population-based approaches to individual approaches and modeling what's relevant to that individual patient. So we've studied pancreatic diseases for a number of reasons. It's biologically one of the most simple organs in the body. It has 3 cell types. They each perform one function, and we have these patients to optimally be managed. So within our platform, we are first facilitating an earlier diagnosis based on the mechanism and not the symptoms or features of a disease. We're using that now with pharmaceutical companies and drug development to help them better stratify patients in clinical trials. And we are using this platform, software as a service, to distribute that knowledge in a clinical decision support manner. We started in pancreatitis, and now we're moving into the overlapping conditions of various forms of diabetes as well as pancreatic cancer. So this is where we generate our primary revenues. We are paid for both the genetic analysis and the clinical decision support packaging that we're delivering to healthcare providers. And this is, I would say, one of the big differentials between a big box genetic store like Ambry or Invitae. And what Ariel does is that most physicians are not trained in how to incorporate genetics into clinical medicine. As I said, a genetic report for a 2-year-old and a 62-year-old is the same, but the clinical needs for a 62-year-old and a 2-year-old are very different. So what this report has kind of generated, this organic traffic from our customer base, is it identifies the mechanisms that are likely failing, what biomarkers should be selected to measure and confirm that dysfunction in that metric, and what are the best practices and therapeutics appropriate for patients like this. And from this, we've not only established a broad customer base at physicians at over 100 institutions across the country. I think we've got over 1,000 patient samples, but this is from a publication that we had earlier this year. We're showing a disease like pancreatitis that was previously had one diagnosis code is actually a combination of factors where you've got statistically common variants that occur that are independently not sufficient to cause disease, unless it co-occurs with a variant that impairs the body's ability to respond to that injury. And it's this 2, 3, 4 hit effect that is driving patients' disease for different reasons. So this has enabled us to model patients into virtual populations, and this is opening up the door for therapeutic optimization. Pancreatitis is a notorious, I would say, graveyard of failed drugs because it was treated as a single disease. And now with our technology and approach, we are enabling earlier identification of the dysfunction and aligning optimal solutions. For pharma, this is really helpful. And where we've been in contact with them is you want to de-risk these assets. By an enriched population, you can get to efficacy endpoints sooner. You also de-risk the asset from being put in the wrong patient type. And the subsequent biomarkers that are selected can be really important when you're trying to evaluate improvement. And ultimately, within our platform, this continues to learn and grow. Our users are leveraging global expertise on complex genetic modeling, systems biology. It is hyperlinked to the most appropriate curated evidence relevant to each patient, so that healthcare practitioner, even if it's one of the first times they're seeing the patient, with this approach, they're able to make the most advanced decisions for each patient at each clinical encounter. Here are some of our customers that we have across the country. And while we've initially designed our models on the pancreas, we see this as essentially the first vertical in the framework to help us start looking at other cell types and other complex organ systems. So the same systemic process that can cause the conversion from fatty pancreas to chronic pancreatitis is a very similar pathology when we look at the liver cell types and the conversion of NAFLD to NASH. And if you think of the interrelatedness of a lot of these systemic dysfunctions, we clinically see patients that also have celiac disease and Crohn's. They're not separate diseases, although they're treated by separate specialists. They're often a systemic dysfunction in a single patient that is causing damage to multiple organ systems. And by this reverse engineering approach, we're able to start being more predictive and more preventative. So summary, we are democratizing access to expert insights in both the endocrine and exocrine pancreas right now. By using this data network and data technology approach, we are enabling a new way to target these mechanistic dysfunctions. And through our clinical decision support platform, we're continually advancing the insights and the therapeutic in alignment for each patient. And I appreciate the opportunity to present today.
Jessica, thank you so much for your presentation, for joining us. I will once again turn it over to Eric and Ethan and Mayank to ask some questions.
Thanks so much, Jessica. Thanks for the presentation, really insightful, very interesting work that you're doing. Just a bit about the workflow. How does a patient get onboarded? When is a good time to do this test? Like when you are approaching the market, what have you seen from your customers? Some information around that. And follow-up would be if it is an FDA-approved test or an LDT right now.
So it's an LDT. We're a CLIA laboratory. We are a dry lab, so we are licensed and able to engage with patients and physicians long-term. The current situation is when a patient presents with symptoms of pancreatitis, We want to help their physician identify why they have this condition. They know that there's something wrong. We want to provide insights to the mechanism. So a physician creates an account on our cloud-based portal. We are telemedicine pre-COVID. They provide the electronic prescription with the patient health data and health summary. The patient gets an invitation to design their own health profile, including quality of life metrics, medications they may be taking. We ship them a buccal swab through the mail that goes to a lab running a proprietary chemistry. And then the clinical evidence-based report is returned back to both the physician and the patient. It includes genetic counseling and all the other elements, but this is kind of the workflow at this point. And we are reimbursed, as I said. We're considered a sole source laboratory because nobody in the country is offering what we're offering the way that we are. And we are paid for both the genetics and the clinical decision support executive summary that comes with each report.
Mhm. Jessica, maybe I can ask, I'm looking at your slide here, and you have basically 3 inputs into your AI system, right? The medical history, genetic information, and patient health profile. How does medical history and the patient health profile differ? And if some of the information is missing, do you get the same quality of output?
Great question. So the medical history is really, as we're kind of, if I go back to this, looking at where the patient is on a disease course. So we have longitudinal data looking at patients like this over 10 years, and we can kind of map out how patients with different combinations of factors progress, how rapidly or severely they progress. So if we know that they've had this disease for 5 years with physical imaging and all these medications, and we're able to look at the quality of life impact on pain, the medications, we can kind of calibrate where they are, because the decisions, I would say the options available to a physician, the earlier we can facilitate a diagnosis before there's chronic scarring and pain syndromes, the better, because that can provide the runway for intervention and alternative treatments.
Yeah. And does the software incorporate other interventions like diet, exercise, drug, behavioral therapy? How does the software handle interventions that preceded the test?
So I would say using pancreatitis as an example, because there had been such little advancement in therapeutic treatments, some of it is anecdotal. We are now actually opening up a new way to identify different treatments for patients based on mechanism. But all of the information, all the publications that are relevant to patients with like this type are actually hyperlinked in our report. So it says, this patient has oxidative stress and CFTR duct dysfunction. We've seen in patients in India, physicians were having success with antioxidants in some patients and they didn't know why. We're now identifying why that is in certain patient types, that they may have a different mechanism that can have an over-the-counter treatment. We're actually in the process now of working and looking at CFTR dysfunction. It actually occurs in about half of our patients. And while these patients don't have cystic fibrosis, they have enough type of dysfunction where it primarily targets the pancreas rather than the lungs. And there's been a lot of work because of Vertex in this space. And there are a number of therapeutic assets that have an opportunity to leverage our database of patients. They've been pre-profiled genetically to meet this mechanistic target. And almost, I would say the majority of our patients have actually consented to be contacted to participate in clinical trials. So they're curated and ready to go for when we would want to roll up a clinical trial.
So for those clinical trials, are you able to tell if it's a fit for the particular mechanism of action, or is it around the compound itself, or is it a phenotypic?
It's a match. So we want to say, so for example, there were some trypsin inhibitors that I think in Japan had shown initial success in some patients, and then the other half they put in the hospital. And there was no visibility into why. It's because the patients that had, I would say, were having success in outcomes had trypsin dysfunction as their primary drivers. And patients that had other dysfunctions that were causing it, if you mess with the trypsin pathway, it compounded those external problems. So what we're doing is looking at the mechanism that's the primary driver of a patient's disease and the mechanism of action for a therapeutic target. When you think of diabetes, so GLP-1s and DP4s carry black box warnings for pancreatitis. Most of our patients clinically at some point will develop diabetes, not because it's type 2 diabetes, because it's diabetes as a consequence of chronic injury and inflammation to the islet cells, because it's in a hostile environment with the local tissue. And those patients, the DLP-1s and GP4s actually exacerbate that and make that severe. I mean, that black box warning could have been mitigated by better patient profiling at the onset in the clinical studies.
And the majority of your revenue to date comes from pharma and clinical trials, or are you in?
Actually, we've just gotten started. We really used the first process of the genetic laboratory and the physicians to establish a database that we have a unique knowledge set around. Long-term relationships, so we're getting longitudinal data on these patients' progression and health profiles, but also because we needed to build the models based on the highest quality data we could. And now we're able to start leveraging that and taking in data from other sources. We've provided services to other genetic laboratories doing whole genome sequencing or arrays because we can impute from a highest quality data set what that data means. So this summer, we actually executed our first contract with a drug development platform. And we actually had a couple of studies funded by AbbVie early on, and now we're looking at leveraging our advanced platform to start doing this, not like a wash, rinse, repeat, but there's a piece of the population for many people.
Thanks.
Jessica, thank you so much for your presentation and for panelists for the questions. Next up, our third presentation is Geneticure, and the co-founder and CEO, Scott Snyder, will be presenting for the company.
Great. Thanks, Stephen. Thanks for having us here from Geneticure. Geneticure is a Mayo Clinic-backed precision medicine company that was initially focused on chronic diseases, specifically pharmacogenomics and hypertension. And we've now built out a pipeline as well as looking at companion diagnostics for device therapies in hypertension. The team comes largely from Mayo Clinic as well. I personally come from Target Corporation, where I ran a large OTC pharmacy business for Target. We have a backbone from Mayo, of course, in both the medical director side and on the business leadership. And our overarching mission is, like others on the panel today, using genetics to eliminate avoidable costs and outcomes in disease, specifically chronic complex diseases, where we use a really unique approach looking at the integrative physiology of genetics to better guide therapy. And we believe hypertension is the biggest opportunity for this technology. It's a good fit for what we do. It's the leading preventable cause of death in the world. It doesn't quite get the attention that it deserves, this silent killer. And we believe that is largely preventable with a more reasonable patient experience, and then also matching unique medicines to unique genetics. The standard of care is pretty broken, so it hasn't changed much over the years, and it usually requires multiple visits back and forth to the clinician. You're titrating and adding medications often that don't work. Each medication has a really well studied bell curve response to therapy. So what may work for you may cause a spike in blood pressure for me. And again, I mentioned that there's a stacking to standard of care. And every time you add a medication, patient non-adherence goes up significantly. And all of these medications can have side effects, either dangerous or annoying, that would cause a patient to stop taking their medication. So when you look at the net result of this, the standard of care, it's really dismal. About 500,000 deaths in the U.S., 131 billion to treat. And with new guidelines, about half the country's adults are considered hypertensive. Very few have it under control. So looking at behind the scenes of what's happening in hypertension, you have a series of genes that cause the organ systems to do what they do. In the cardiac system, you have genes that regulate heart rate, heart contractility. You have a whole bevy of medications used to treat that. In the cardiac system, same in the renal system, you have sodium reabsorption that's regulated by genetics, and same with the vasculature. So generally, we built a panel of genes across these three organ systems, and then applied a complex algorithm to the panel, weighting the particular genes in each of the organ systems against each other. And then also the organ systems themselves kind of duke it out in the algorithm based on functionality of the SNPs and also the strength of the peer-reviewed literature behind each of them. The result then is Geneticure for Hypertension Pharmacogenomics. It's a buccal swab collection as well. It's a one-page report that guides towards a ranked recommendation of medication classes. Again, we also believe that clinicians, this is meant to be read on the way into the consult. It groups the organ systems together and then shows functionality with each of the SNPs, but then also the organ systems themselves. We took a very much evidence-first approach to building Geneticure. The first 5 years of our existence were heads down in clinical trials. We're now starting to exit that phase into commercialization. But the results of the trials have been very positive. So we have 3 completed on about 700 subjects. This is 2 retrospective trials and then a pilot RCT. We demonstrated that we can control almost all patients within a very short time period, 97%. The big outcome variable here is that patients that, and this was a retrospective design, patients that would have matched Geneticure's first recommendation for a therapy would have 36% lower blood pressure than those who did not match. And then that, of course, drives down the adverse event rate. A blood pressure reduction of this size is equivalent to anywhere between a 40 and 60% reduction in adverse events like stroke, heart attack, cardiovascular disease. Those are the events that really drive the cost model, which has also been peer reviewed and published. We're about half the cost of standard of care, and it really comes from changing the mix from hypertensive uncontrolled to hypertensive and controlled. So we're really unique in pharmacogenomics and now looking beyond into companion diagnostic for device, but there's a wide variety of, a large number of companies out there looking at the way drugs are metabolized into the bloodstream by analyzing markers like CYP2D6, CYP2C19. These have some great applications, but when it comes to complex chronic diseases, drug metabolism alone is essentially meaningless from an outcome standpoint. In hypertension, I can have 20-fold the amount of a beta blocker circulating in the blood plasma volume, but without a functional drug target, that beta blocker will be meaningless. So we are an inch wide, mile deep into each disease. And then I mentioned evidence first, and we also have strong IP. We have patents awarded on our pharmacogenomics panel for hypertension in the US and the EU. That's Geneticure, and look forward to your questions.
Scott, thank you so much. I'll turn it over to the panelists.
Scott, thank you. Eric Messner here. How do you see the output of your test, your screen, meshing with or contradicting, hypertension guidelines, whether from American College of Cardiology or International Society of Hypertension? They're, at least in my understanding, kind of the typical algorithm of drugs that are tried. And how do you see that in your go-to-market overlapping, or if they contradict, how do you plan to work through that?
Yeah, we are seeing them contradict. And the interesting thing and background on that, as you probably know, is those guidelines were kind of based on population averages, right? There's really no, there's JNC8 guidelines and ACC guidelines. When you go health system to health system, or even a physician to physician, they all follow their own unique algorithms and best practices and favorite medications. Oftentimes when we ask physicians why they start where they start, we really don't get a good answer. It's just kind of like, I generally have seen success with that, et cetera. And there's certain considerations for comorbidities as well, which is why in our report, we provide this ranked recommendation, because there may be a contraindication to another disease, for example. But we believe, it's evidence, and we built a really, we're punching way above our weight in terms of peer-reviewed evidence. We now have 5 or 6 manuscripts published both clinically and economically We're planning this large RCT, which will be kicking off probably late Q1 next year. It should only take about 6 months for an initial readout, and then we'll publish. And we just believe that, based on the strong scientific and clinical backbone of our team, that clinicians are done with the promise of precision medicine without the peer-reviewed evidence. And so we just think that it will take time, but we'll change those guidelines over time. There hasn't been anything in our clinical evidence to date that says we're not on the right track.
Thanks, Scott. This is Mayank, by the way. So just a quick question around reimbursement. One of the challenges that ambulatory tests face is reimbursement, and the adoption into the primary care space, again, speaks to that. So can you just describe how are you approaching that particular problem here, and when do you have a better line of sight into reimbursement on all of this?
Yeah, absolutely. So right now we're targeting $249 for our test. We've published evidence in the Journal of Medical Economics that $249 returns 16x ROI to payers inside of 3 years. So it's all built on payer ROI. We also have pretty healthy margins at $249. So if we need to stress test that price for even higher ROIs, we can do that. That was all built based on clinical data into health economic data. So based on the reductions that we're showing in blood pressure versus standard of care, we know incidence rates and all of those expensive adverse events go down significantly. So there's some contribution in the model to reduced office visits and medications, but really the driver of that is well-studied reductions in expensive adverse events for payers that drives the model. So we'll probably, we've had a good response from payers thus far. We haven't formally approached them until we get through our next RCT, where we'll take the payers for widespread coverage and also breakthrough device designation.
You mentioned earlier you're half the cost of standard of care. What do you see as the standard of care right now?
Well, it's really that well-studied, the number of visits to reach control, the number of prescriptions per patient, and then the incidence rates around adverse events, which are all well published. And all of these are cited in the model and in the publication, but that's what we use to build the model. And like I just said, based on what control rates are today and in our clinical work versus what we've been shown to reduce them by, we can then surmise, and it's really a simulation, what the decrease in cost is from those events.
Is there a trigger that where the clinician would say, okay, go get this test, get the genetic test done and run through the program? Is it just high blood pressure, or is there something else?
Well, of course, long term, we believe it should be all high blood pressure. The decision tree should go. You're diagnosed on usually a confirmatory visit. You're not gonna the first reading. You'll be sent home for TLC, therapeutic lifestyle changes. Then when a second reading is confirmed, you'll order, we believe long term, this is 5-10 years down the road, the Geneticure for renal denervation screening. Because if you're a candidate for renal denervation and that therapy has shown promise on mild and moderate hypertensives, there's no reason you shouldn't have that therapy. But if you're not a candidate for that, you get the Geneticure for pharmacogenomics test. The way it works today is system by system. We're working our way into kind of digital formularies within care algorithms. But it depends on how the systems want to treat it. Some are attacking their most severe cases first. Some want to approach rural populations because this can all be done remotely with an at-home cheek swab. So it just depends.
And then a lot of these patients end up having other, comorbid activities and whatever else. Are you guys keeping the genetic data once they've been sequenced, or will we have the information for the report and you throw the rest of the genomic information out?
We destroy the DNA today. Other than our clinical work, we just feel that that's kind of where the environment is and what patients are expecting. And we also, we don't have additional applications beyond hypertension that we want that are market ready. We have a lot in development, but they aren't market ready. At some point, your DNA will be on file, and we'll there's companies that are doing exome plus sequencing and will ping the database in the areas that we want. None of them have been able to have good clinical coverage on the SNPs that we analyze. We have a lot of rare variants and upstream indoles and things like that. But we do believe that's where it will be eventually.
Mm-hmm. And Scott, you're raising money now to, I assume, to commercialize the hypertension test. And in the absence of a reimbursement, a payer reimbursement, what is your go-to-market strategy off the bat?
Well, there's, outside of widespread and CMS reimbursement, there are plenty of organizations that are interested in paying for the test. Those are typically those organizations that have line of sight to the cost of care. So payer provider organizations. We're talking to a very large payer right now who has one of the largest clinical networks as well. And these, based on the evidence that we've shown, there's no reason why these shouldn't be covered by those systems. So outside of like a widespread CMS coverage decision, you also have, given the prevalence of the disease, quite a few centers of excellence around hypertension, academic medical centers that we're working with that are centers of excellence for hypertension, concierge medical practices that want to offer the latest and greatest technology. If it were a rare disease, these would be less appealing go-to-market channels, but because it affects roughly half of adults, all of these channels are appealing.
Mm-hmm.
Scott, thank you so much for your presentation. Thanks again, Jessica and Eric, for your presentations. Thanks to our panelists, Ethan and Mayank and Eric, for your great questions, and to everyone for joining us. If you'd like to connect with any of the companies and didn't capture their information, please let us know. You will receive an email with a survey, so please fill that out, provide some feedback, both for us as well as any feedback you'd be interested in sharing with the companies. If you signed up again for the virtual networking, look in your email for a note from Kay Nietzkaj from Matter, and there will be a link there to click to go to just at the conclusion of this program. This program is our last public forum of 2020. We've got some great things planned for 2021, and of course, we're looking forward to a much brighter year for our country and for the world. So you can learn about all of our upcoming programs on our website, matter.health. I wish you all a great holiday, happy new year, and a terrific rest of your day.